are very, very important. I told you hormones are auxins and cytokinins as we discussed earlier. Then next, you have the environmental factors. Environmental factors is like light, temperature, then photo period. అంటే ఏంటి మనకి మనము photosynthesis లో చదువుతుంటాం అనమాట. What we study in the photosynthesis, you have the dark phase and the light phase you have. Same here also, you have the photo period you have. So plants have to get kept in the dark phase and in the light phase. Annitkanటే important sterile conditions. The sterile conditions have to be maintained from the day one till the plant is taken into the field. Okay. Then you have the media. What type of media you are using? Different types of medias are there. What type of media you are using? That media is very, very important. Next is selection of the explant. Explant ante enti? I told you in the first slide lo చెప్పాను. In the first slide, what did we discuss? It is any type, any part of the plant anamata. Seeds gani, leaf disc gani, meristems gani, apical meristems gani, protoplast, tissue, cells, embryos, anything. Those are called as the explants. But in that explant selection also is very, very important. So enti adi, if it is younger and if it has more of meristematic activity, then yeah, the explant will be fantastic. But if it is what you call older plants, or if the selection is not suitable, that means you have certain plants like woody plants, you have you have you have woody plants, you have some other what do you call older leaves, you have all those things like that you have. So the success rate of the tissue culture manaki తగ్గిపోతుంది అనమాట. నెక్స్ట్ వీ హ్యావ్ టు డెఫినెట్లీ సీ ద జెనెటిక్స్ ఆఫ్ ద ప్లాంట్. మనం ఏం తీసుకుంటున్నాము అనేసి ద జెనెటిక్స్ ఆఫ్ ప్లాంట్స్ ఇస్ very, very important. Based on the genetics only, manaki next tissue culture plant nunchi whether you will be getting flowering vastunda rada, a haploid adi annadi, all those things are dependent on the genetics. That we will see next manaku slides vastunte aa slides lo chudochu manam. Okay, so next tissue culture dantlopali you have I have told you the factors. Apart from the factors, manam three important fundamental ability ఒక ప్లాంట్ హ్యాస్ సర్టన్ ఎబిలిటీస్ అనమాట. ఎట్లా మనిషికి ఒక మాట్లాడే ఎబిలిటీ ఉంటుంది, మనిషికి తిట్టే కొట్టే ఎబిలిటీ ఉంటుంది. సేమ్ లైక్ దట్ ద ప్లాంట్ ఆల్సో హ్యాస్ సమ్ ఫంటాస్టిక్ ఎబిలిటీస్ ఉంటాయి అనమాట. ఏంటి అంటే ద ఫస్ట్ వన్ ఐ టోల్డ్ యూ టోటీ పొటెన్సీ. టోటీ పొటెన్సీ ఈస్ ఏంటి అంటే టోటీ ఇస్ టోటల్, పొటెన్సీ అంటే రీజువినేషన్ అనే అర్థం అనమాట. అంటే ప్లాంట్ నుంచి యూ టేక్ ఎనీ స్మాల్ పార్ట్ విచ్ ఇస్ కాల్డ్ ఎస్ ఎక్స్ప్లాంట్ అనేసి అని అన్నాం అనమాట. ఏదన్నా సీడ్ కానీ, ఎంబ్రియో కానీ, టిష్యూ కానీ, సెల్ కానీ, ప్రోటోప్లాస్ట్, యూ టేక్ దట్ స్మాల్ వన్ and you put it in the nutrient media along with the hormones under sterile conditions, you will be getting the total plant. You will be getting the total plant. That capability of the plant to produce a total plant from a single cell or single explant is called as totipotency. Okay, totipotency ante enti? From a single explant or from a single cell under sterile conditions, hormones and nutrient media, it can produce or it can establish total plant, full plant. That capability of the plant is called as totipotency, but is not there in the animal system. In the animal system or in the organisms, nowadays, you can see the liver transplantation. But the liver transplantation also, it is in vivo. Ante manishi lopalne manaki you can have the liver transplantation. So if you are transferring the liver from the donor to the plant, so many matchings have to be done and you have to put it under in vivo condition. Ante manishi lopalne you have to do that anamata. You just can't take the liver cells and you can't culture it outside. But you have nowadays umbilical cord లోపల stem cells వస్తుంది. Stem cells work is going on very fantastically, but it is costing so much. It costs crores and crores of rupees. Still, people are saving the stem cells. The rich people people or the elite people are saving the stem cells, but for that saving also, for that rental purpose only, they have to pay some lakhs and lakhs of rupees. So the tissue culture thing in the animal system is lagging behind due to its capability of having either meristematic activity or the cell division activity or totipotency activity. ఇవన్నీ అందులో ల్యాక్ అవుతుంది అనమాట, విచ్ ఇస్ యూ ఫైండ్ దట్ ఎబిలిటీ, ఫంటాస్టిక్ ఎబిలిటీ ఇన్ ద ప్లాంట్ సిస్టమ్. నెక్స్ట్ యూ హ్యావ్ ద డీడిఫరెన్షియేషన్ మనకి కనిపిస్తుంది అనమాట. సో వాట్ ఈస్ దిస్ డీడిఫరెన్షియేషన్? you're taking some explant in a root shoot disc and anything you're taking, that shoot disc again, it is becoming into meristematic condition anamata. Adi meristematic meristematic అయిపోయేసి it will develop into total plant anamata. Ante that is a big virtue the plant is having anamata. Enti ante ippudu manam you take the plant, usually you will have where all meristems will be there, you will be having intercalary meristems or the apical meristems you will be having here, but or axillary meristems you will be having. But what we are telling here in tissue culture, you take any part, you take tissue తీసుకోండి, cell తీసుకోండి, leaf disc తీసుకోండి, when you put that in the artificial media, దానికి meristematic activity వస్తుందన్నమాట. It will get the meristematic activity అన్నమాట. So that fantastic activity we find in the plant system. So that is called as a redifferentiation. Again, from that you will be getting redifferentiation, and that meristems will go into the whole plant. So tissue culture is nothing but it is a capability of having redifferentiation and redifferentiation. D and re అంటే ప్లాంట్ నుంచి అది మెరిస్టమ్ అవుతుంది, మెరిస్టమ్ నుంచి అగైన్ అది ప్లాంట్ అవుతుంది. సో దట్ ఈస్ కాల్డ్ as dedifferentiation and redifferentiation, and that total thing is called as totipotency of the plant anamata. So these things have to be kept in mind anamata. So next, when you come to the history of the plant tissue culture, so direct మనకి plant tissue culture వచ్చేసేలేదన్నమాట. So many scientists, so many things they have worked, from that continuously working we have came into this plant tissue culture anamata. So when you see here, first మనకి Sheldon and Shawn, they have given us a cell theory, they have given anamata. In the cell theory itself, they have given very important things like ఒక సెల్ నుంచి ఇంకొక సెల్ వస్తుంది. సో ఫ్రమ్ ద ప్రీవియస్ ఎగ్జిస్టింగ్ సెల్ ఓన్లీ న్యూ సెల్స్ ఆర్ ఫార్మ్డ్ అనేసి అని మనం చెప్పుకుంటాం అనమాట. సో దేర్ ఇట్ సెల్ఫ్ ఇట్ షోస్ దట్ ఇట్ హ్యాస్ అ పొటెన్షియల్ టు గివ్ న్యూ సెల్స్. ఓకే, ఇట్ హ్యాస్ పొటెన్షియల్ టు గివ్ న్యూ సెల్స్ అనేసి. నెక్స్ట్ ఇన్ 1908 we had Herman Land. He has done the first attempt of plant tissue culture. Then in 1939, here we have a scientist called as White. We have White's medium also. He has discovered a medium called as a White medium, and he had discovered the callus culture. That means from the explant, he had developed the growing callus culture he has discovered. Then in 1946, we have scientist called as Ball. He had developed the whole plant from the shoot tip. Shoot tip lo emuntundi manaki, in the shoot tip we will be having the meristematic activity. So this Ball, he has taken shoot tip as a explant. From that he has developed the whole plant in way back in 1946. Then in 1950, the same scientist Ball, what we have discussed here, White has discovered the callus culturing he has discovered. But what Ball has done in the 1950s, he has taken callus, and from the callus he had developed the organs and shoot system and root system he has developed anamata. Next, Muir in 1954, he has developed a plant from a single cell, kevalam oka single kanamu, eka kanamu toti he has developed the whole plant. Then ప్రోటోప్లాస్ట్ ఐసోలేషన్ అంటే మనకి ఒక కణము లోపల ఏదైతే సైటోప్లాజం సెల్ వాల్ తీసేసిన తర్వాత మనకి ఏదైతే మనకి లోపల సెల్ మెంబ్రెయిన్ లోపల ప్రోటోప్లాస్ట్ ఉంటుందో ఆ ప్రోటోప్లాస్ట్ ఐసోలేషన్ ఫస్ట్ టైం కాకింగ్ అన్న శాస్త్రవేత్త, he had done the protoplast isolation. Okay, Next, when you come again here, MS media అని మనం చదువుతూ ఉంటాం అనమాట. So this MS media was given by Murashid and Soog. So under their name, we call that medium as MS media. So one thing you have to remember, students, what you have to remember is మనము టిష్యూ కల్చర్ చేసేటప్పుడు you say మనం MS media యూస్ చేస్తున్నాం, వైట్స్ మీడియా యూస్ చేస్తున్నాం, LS media. So that is just the basic nutrient media. But అందులో మనము హార్మోన్స్ ఎంత కాన్సన్ట్రేషన్ వేస్తాము అన్నది ఇట్ డిపెండ్స్ ఆన్ యువర్ standardization. So Murashi and Shrew or Blades media or Whites media or LS media, any type of media, those are just the different types of media which we use. And in that we have to add the concentration of hormones what is required for that particular plant అన్నమాట. సో ఇట్లనే మనకి we have angiosperms, we have gymnosperms, we have dicot plants, we have monocot plants, we have forest plants. So each one, before forest plants or the tree plants, tissue culture was very, very difficult. But nowadays we are doing lot of tissue culture plants in the forest plants also. That is tree species also they are doing. Okay. So next, clonal propagation, or in orchids, clonal propagation was done by Morel in 1964. Then haploids were developed by Guha in 1964. Haploid means pollen grain. Pollen grain అంటే it's a male gamete. From the male gamete, they have developed haploid plants అన్నమాట. Only male. అంటే only from the pollen grain. There is no union. There is no fertilization between the pollen and the ovule. There was no fertilization. So from that, they have developed haploid plants. Then in 1970, power, this scientist, she had done fusion of protoplast. సో మీరు అందరు అనుకుంటున్నారు ఏంటి ప్రోటోప్లాస్ట్, ఎందుకు ఈ ఫ్యూజన్, ఎందుకు హ్యాప్లాయిడ్స్. బట్ ఈచ్ వన్ హిట్ హ్యాస్ ఇట్స్ ఓన్ ఇంపార్టెన్స్. హ్యాప్లాయిడ్స్ లో తీసుకుంటే యూ విల్ బి హ్యావింగ్ వన్ ఇంపార్టెన్స్, ఫ్యూజన్ ఆఫ్ ప్రోటోప్లాస్ట్ తీసుకుంటే యూ విల్ బి హ్యావింగ్ వన్ మోర్ ఇంపార్టెన్స్. దట్ సేమ్ క్లోనల్ ప్రాపగేషన్ తీసుకుంటే విల్ బి హ్యావింగ్, ఈచ్ వన్ విల్ have its own prominence, each one will have its own applications. For example, in chapter haploids are there. So when it is haploid plants, what happens is you don't have the two things, and ovular and pollen grain fertilization has not happened. Suppose if fertilization happens, what happens? You will be having embryo development. From the embryo, what you will get? You will be getting the seeds. So haploids లోపల ఏమవుతుంది అంటే వాట్ దే డూ ఇస్ కాల్చిసిన్ ట్రీట్మెంట్ ఇచ్చేసి దే విల్ డూ ద వాట్ యు కాల్ దే విల్ డబుల్ ద chromosome and you will be getting the plant anamata. So దాంట్లోపల కూడా if you get fruits, what will happen is you will be getting the seedless variety of fruits మనకి వస్తుంది అనమాట. So you have the seedless plants, or you if it is haploids, you will be having different types of what you call a leaf developments లోపల different characteristics అట్లాంటివి you will be finding. Suppose protoplast fusion and you take two different plants, one will be having disease resistance, another will be having high yield anamata. So you take both the protoplast, you fuse both of them, so it will become a single protoplast. So a single protoplast, you will grow the plants. So what all characteristics you will get? You will get both the characteristics you will get. You will be getting the disease resistance and you will be getting the high yield, both the things you will be getting there. So each one, it has its own what you call applications it has towards mankind or for the development of the science. Okay. See, then in vitro cultures as అనుకున్నాం. What are the in vitro cultures? Ante what are the explants we can use ani that we'll be discussing. So first is a culture from the intact plants. Ante you can take seeds or seedlings cultures you can take. Then embryo. So embryo always it should be immature embryo anamata. So if you take the immature embryo, what happens is you will be having little bit of what you call meristematic activity మనకు అందులో కనిపిస్తూ ఉంటుంది అనమాట. So you can take the immature embryo, you can take. Then you can do organ culture. In the organ culture, you have the shoot tip culture, you have the root culture, you have the leaf culture, and the anther culture. So many cultures are there. Then again, you have the Callus culture. I told you from the explant can either directly get the root system and the shoot system, or in between you can have a stage called as a calcing stage. So callus culture also you can do. Then cell suspensions, usually for the secondary metabolite productions and all, you'll be having this cell suspension cultures another. After that, you have the protoplast culture. So many different types of cultures are there. All these cultures are done under in vitro conditions. In vitro conditions means, keep in mind, it is under the laboratory conditions. Laboratory conditions and it has to be having the nutrient media undali, it should have the hormone system, and over all the above, it should be under the sterile conditions. Okay. Next, you have few applications of the tissue culture. One is the micropropagation. Second is the germ plus preservation. Then you have somochlonal variation, dihaploid production. I told you the haploids from doubling JC, you'll get the seedless variety. Then you have the protoplast fusion. Then secondary metabolite production, cell suspension cultures of callus suspension cultures. Until you have the secondary metabolites, so many metabolites plants will be producing, that you will be having. Then genetic engineering lopala, after you make the vector construction చేసిన తర్వాత you put that particular vector into the plant, either through agrobacterium or through the bioride gun. And how you develop the plants, you develop through the tissue culture method only you will be developing. So you have so many different applications. All these applications మనం నెక్స్ట్ వచ్చే క్లాసెస్ లోపల ఒక్కొక్కటి we will be studying in detail అన్నమాట. Next, first we'll come to the micro propagation. So micro propagation is nothing. The name indicates micro అంటే చిన్న దాని నుంచి ప్రాపగేషన్ చేయడం అన్నమాట. అంటే ఎక్కువ ప్రాపగేషన్ చేయడము. From small you can do the large type of propagation. దాన్ని మనము మైక్రో ప్రాపగేషన్ అంటాం. For example, I will ask you a question. ఇప్పుడు we are taking seeds. Okay, you take ఒక వడ్ల గింజ అన్న తీసుకోండి, or you take చింత గింజ అన్న తీసుకోండి, or you take పల్లీలు గ్రౌండ్ నట్ బుడ్డలు అన్న తీసుకోండి. If you put one in the soil, in the suitable soil, how many plants we will get? ఒక్క వడ్ల గింజ నుంచి మనకి ఒకే ఒక రైస్ మొక్క వస్తుంది, ఒకే ఒక పీనట్ గ్రౌండ్ నట్ నుంచి ఒకే ఒక ప్లాంట్ వస్తుంది, ఒకటే ఒక చింతకాయ గింజతోటి ఒకే ఒక ప్లాంట్ వస్తుంది. But here, when you are using the tissue culture, what you have is you are having the stages in చెప్పాను కదా. From the explant, you will be getting the callus అనేసి అని చెప్తున్నాను. From a single callus, you can have about 400 to 500 plants ని మనము డెవలప్ చేయొచ్చు. From a single callus plate, you can develop 400 to 500 plants ని మనం చేయొచ్చు. అంటే from a single small disc, you will be able to produce so many plants అన్నమాట. That is called as a micro propagation. అంటే at the small level, under the laboratory condition, using a very small explant through callus medium, you can develop so many n number of ప్లాంట్స్ ని మనము ప్రొడ్యూస్ చేయొచ్చు. ఇప్పుడు చూస్తున్న బయట నారు కానివ్వండి, ఇప్పుడు చూస్తున్న బయట ప్లాంట్స్ కానివ్వండి, ఐదర్ ఇట్ కెన్ బి బనానా, ఇట్ కెన్ బి ఎనీ ప్లాంట్, వంకాయ కానివ్వండి, సో మెనీ ప్లాంట్స్ ఆర్ దే. ఆల్ దోస్ ప్లాంట్స్ ఆర్ టిష్యూ కల్చర్డ్ ప్లాంట్స్ అనమాట. ఎవ్రీథింగ్ దే ఆర్ టిష్యూ కల్చర్డ్ ప్లాంట్స్ ఓన్లీ. So that's why they are selling it for 1 rupee, 2 rupees, like that they are selling outside. So in this micro propagation, how you can do, and a micro propagation also you will be doing, no matter how you can do so many methods in the first is embryogenesis. And embryogenesis means it looks like embryos, but they are not direct embryos. They are not embryos formed through the fertilization. That you must remember here. So embryogenesis is direct embryogenesis or indirect embryogenesis, that I will tell you. Next, organogenesis, that is organs will come via the callus formation. Or you have direct adventitious and thick adventitious roots at last, like that you will have the organs formation, you will be getting directly. Then you have micro cutting. So you have meristems and the shoot tips or the bud cultures untadi, then micro cutting stage kuda you can put in the culture media, you will be getting lot of plants. So first we will see embryogenesis. I told you embryogenesis, so first one is the somatic embryogenesis. So in the లోపల somatic embryogenesis is, mind you, the word here somatic అనేసరి మనం చూసుకోవాలి. Somatic is always the vegetative thing anamata. It is not, you have two types of cells in the body, somatic cells and the germline cells anamata. Germline cells లో meiosis అవుతుంది, that is used for reproduction కి అది యూజ్ అవుతుంది అనమాట. వేరే అదర్ హోల్ బాడీ, ఐదర్ హ్యూమన్ బీయింగ్స్ కానివ్వండి, యానిమల్ సిస్టమ్ కానివ్వండి, ప్లాంట్ సిస్టమ్, రిమెంబర్ ఇట్ ఈస్ somatic cells only. So here somatic cells lopare you don't have union of pollen and the ovule. Manaki fertilization undadu. That you have to remember. Tissue culture, you don't have union of the male and the female gametes. Directly you'll be getting the plant system. That's why you call that as somatic embryogenesis. So here you will be having here directly somatic embryos are formed here, or they are non-germ cells. That means they don't come under the reproductive cells, that is the germline cells. So usually they are formed in the callus intermediate stage, callus locally you will get. So there are so many like things, heart-shaped things here. So all these are somatic embryos. So you can see here n number of things. You take them and you put it in the media, so many will come. But ఒక seed తీసుకొని if you put, only one plant will come. But same callus లోపల see, so many somatic embryos are there. So many plants will come here. Okay, so this is called as somatic because there is the formation of embryos are formed from the somatic cells, not from the germline cells. See, somatic embryogenesis. Explant తీసుకోవాలి. From the explant you put it, you will get the callus. Callus lopalanta like this. So many embryos will come here. See, the embryos will look like this. You take out all the embryos and you put them in the medium. When you put them in the media, you will be getting the plants మనకి ఇట్లా వస్తుంది అన్నమాట. Okay, so this is in the peanut. Peanut అంటే గ్రౌండ్ నట్స్ లోపల. సో గ్రౌండ్ నట్ లోపల మనము ఏం చేస్తాం, మనం ఒక పల్లి తీసుకుంటే యు గెట్ ఓన్లీ వన్ ప్లాంట్. బట్ హియర్ వాట్ యూఆర్ డూయింగ్, యూఆర్ టేకింగ్ ద సొమాటిక్ ఎంబ్రియోస్ తీసుకొని క్యాలస్ పైన ఈ సొమాటిక్ ఎంబ్రియోస్ ని మనం యూఆర్ సపరేటింగ్ ఇట్ అండ్ యూఆర్ పుట్టింగ్. సో ఇవన్నీ ఉన్నాయో సీ సమ్ 400, 500, ఆల్ దట్ యూ డూ కల్చరింగ్, యూ గెట్ సో మెనీ ప్లాంట్స్ యూ విల్ గెట్ హియర్. Next is the organogenesis. In this organogenesis, as the name indicates, organs ఏ మనకి డైరెక్ట్ గా ఫామ్ అవుతుంది అనమాట. అంటే డైరెక్ట్ గా ఎక్స్ప్లాంట్ ఒక్కొక్కసారి మనకి డైరెక్ట్లీ ఫ్రమ్ ద ఎక్స్ప్లాంట్స్ యూ విల్ బి గెటింగ్ ద రూట్ సిస్టమ్ అండ్ ద షూట్ సిస్టమ్, ఆర్ క్యాలస్ వచ్చి క్యాలస్ నుంచి కూడా యూ విల్ బి గెటింగ్ ద రూట్ అండ్ ద షూట్ సిస్టమ్ అనమాట. బట్ వాట్ ఎవర్ ఇట్ మే బి, యూ విల్ బి హ్యావింగ్ ద ప్రొడక్షన్ ఆఫ్ ద roots and the shoots or the leaves మనకి డైరెక్ట్ గా వస్తుంది అనమాట. సో అది ఇట్ కెన్ కమ్ డైరెక్ట్లీ ఫ్రమ్ ద వాట్ యు కాల్ ఎక్స్ప్లాంట్ అంటే ప్రీ ఎగ్జిస్టింగ్ మెరిస్టమ్స్ అంటాం, ఆర్ వితౌట్ డిఫరెన్షియేషన్ అంటే ఎక్స్ప్లాంట్ డైరెక్ట్ గా ఇట్ కెన్ కమ్, ఆర్ ఇట్ కెన్ ఇన్వాల్వ్ ఎంబ్రియోజెనసిస్ త్రూ callus also it can occur. Okay, see these are the direct embryogenesis, direct organogenesis. అంటే you take the explant you put the suitable hormones. In that suitable hormones you will be getting this particular thing anamata. You will be getting the plants మనకు వచ్చేస్తాయి. So here shoots are there. Next you take it, you put it in the rooting hormone, roots will be formed anamata. Okay, so somatic embryogenesis and organogenesis, for both these methods it can be used for the micropropagation మనము యూజ్ చేయొచ్చు అనమాట. Okay, then this micro cutting. So what is this micropropagation? So micropropagation is nothing but it is just a science, or it is a way of doing multiplication of plants under in vitro conditions. Adoka art anamata. So based on the science, based on our technology, what we are doing, we are playing with the plants. We are producing multiple plants from a single explant, under the laboratory conditions, which is called as a in vitro conditions. That's the usually what you have. Usually you will be getting the meristems with or without the callus stage manaki untundi anamata. So from that you will be getting the plants manaki vastundannamata, as I have showed you earlier. Okay, So what are the steps? So first you have to select the mother plant ని మనం select చేసుకోవాలి. After selecting the mother plant, you have to go for sterilizations. After the sterilization, you put that in the initial growth media. What I told you, initial growth media, both hormones will be equal. That means the auxins and the cytokinins will be under the same ratio. Then next you have to go for multiplication. So for that, what you'll do, you'll take the X plant and first you will put putting it in the shooting media. In the shooting media, the cytokinin will be more when compared to the auxins and matter. Next, you will be going to the third stage, that is the rooting. Until after the shoots are formed, you put them in the rooting media. Then here the roots will be formed. After the roots are formed, what you do? You take this X plants and you put it in the small vermiculite, not vermicompost. Vermiculite will be there in the laboratory condition only. After putting that, after its certain growth, you put that in the sterile soil in the laboratory condition only. That is called as hardening. So after that, you will take it and you will put it in the what you call soil. You will be putting, or you will put it in the glass house or the net house. You will put. After that only, you will be taking it to the field level. See like this. See, you're taking the meristem part, meristem or the shoot tip they are taking. So we have to put the apical meristem in the media. Then from that you will be getting the what you call shoot system. Then after the shoot system, you put that for the root system, rooting hormone. Then from that you will be getting the whole plant. You transfer it into the sterile plant. Okay. So what? Why is micropropagation important? Enduku micropropagation important? Now you it has so many advantages. It has. So you from first one is I told you, from a single plant you, from a single seed you'll getting a single plant. But for the business purpose or for the development of science and or for the business and what you'll do, you will do the micropropagation. So from single X plant you will get 100 plants. So you take 10x plants, how many plants you'll get? 400 plants you'll be getting. So that 400 plants, if you take at least to the field level, at least 200 plants or 300 plants will survive for us. So that is you're propagating it and you're selling it for the more cost. So that is one basic thing you're having here. Basic thing is for the business purpose and also for the research purpose, we can say. But what features it has? ఎందుకు మనం micropropagation కి వెళ్ళాలి? What features it has? First one is clonal reproduction. So clonal reproduction అంటే ఏంటి అంటే always when human beings are there or the organs are there, it should maintain heterozygosity. అంటే మనకి టూ జీన్స్, మనకి టూ సెట్స్ ఆఫ్ మనకి డిప్లాయిడ్, అంటే వన్ విల్ బి గెటింగ్ ఫ్రమ్ ద మదర్, వన్ విల్ బి గెటింగ్ ఫ్రమ్ ద ఫాదర్ అనమాట. సో బోత్ ఇట్ బికమ్స్ హెటరోజైగస్. నా ఇఫ్ సపోజ్ మదర్ దే రెండు సార్లు క్రోమోజోమ్స్ వచ్చినాయి అనుకోండి, వాట్ ఇట్ విల్ బికమ్ హోమోజైగస్ అనమాట. సో హోమోజైగస్ లోపల ద వయబిలిటీ విల్ బికమ్ less and less. The hybrid vigor or the other disease resistance or any other characteristics will increase as the heterozygosity increases. So the clonal reproduction will be maintained by this heterozygosity in the micropropagation. That is one future. Next, in this you will be having many multiple stages. You can recycle it so many times with unlimited clones మనం చేసుకోవచ్చు అన్నమాట. So many clones you can do. So mostly what you can do it for the commercially for ornamental plants, లేకపోతే వెజిటేటివ్ ప్రాపగేటెడ్ ప్లాంట్స్. సో మెనీ ఆర్కిడ్స్ ఇప్పుడు బయట వెనకటికి యూ డింట్ గెట్ వన్ ఆర్కిడ్ ఆల్సో. ఆర్కిడ్స్ వేర్ వెరీ వెరీ రేర్ టు సీ అనమాట. యూ నెవర్ యూస్ టు గెట్ యాంథూరియమ్స్, యూ నెవర్ యూస్ టు గెట్ జెర్బెరా, సో మెనీ. బట్ నౌ డేస్ బికాస్ of this చామంతి కానివ్వండి, క్రిజాంతమం కానివ్వండి, or you have the orchid plus కానివ్వండి, so many plants we are getting or ornamental plants so many are getting they are cloning and they are getting micro propagations. Then it's easy to manipulate and productions అనమాట. అంటే easily you can do the manipulations అనమాట. అంటే ఫీల్డ్ సీజన్స్, ఎన్వైరన్మెంటల్ ఇన్ఫ్లుయెన్సెస్ అవంతా ఏమి ఉండదన్నమాట. You don't see all those things in the micropropagation. Why? Because you have standardized the procedure. According to standardized procedure, you will be doing it in the laboratory conditions. Finally, it will go out అన్నమాట. So when it goes out, it can maintain hardening ఒకసారి it is done in the laboratory conditions. Outside, it can survive very easily. Next, one more thing important here, what you have to do is most of the time micropropagations they will be doing with the apical meristems tissue. Apical meristem tissue lopala you will be having the meristems. Meristems always they are disease free. Meristems always they are disease free anmata. Just because you're culturing, you're doing the whole tissue culture with the shoot meristems or the apical meristems, the plants which are produced will have that meristems characteristics it will have anamata. So naturally, when the apical meristem is మనకు అది డిజీజ్ రెసిస్టెంట్ ఉంది, దానికి డిజీజెస్ రావు, వైరస్ ఫ్రీ అండ్ డిజీజ్ ఫ్రీ అనుకోండి. సో naturally all the plants which are produced also will be disease free అన్నమాట. సో డిజీజ్ ఫ్రీ ప్లాంట్స్ క్యాన్ బి ప్రొడ్యూస్డ్. మనకి ఇప్పుడు ఎస్ఏ టైప్ క్వశ్చన్స్ యుల్ బి గెటింగ్ అనమాట. హౌ టు గెట్ డిజీజ్ ఫ్రీ ప్లాంట్స్ through tissue culture and it is through you will be using the shoot meristems or the apical meristems. Through that you will be getting the disease free plants అనమాట. Suppose if the meristems లోపల diseases వచ్చినాయి అనుకోండి, ఏమవుతుంది, the plant will not grow at all. The plant will not grow at all. It will not have the capability anamata. It meristems itself will have rejuvenating cytoplasm. It will have all the enzymes. All it will be having it is because it's just growing cells. Kabatti all rejuvenating things will be there. There you will never get in any of the meristems. Intercalary meristem కానివ్వండి, axillary meristem కానివ్వండి, apical meristem కానివ్వండి. In the meristems you will not get diseases at all. Okay. Next, we have the embryo culture. So, embryo culture, you can do it by rescuing certain embryos. Okay. Suppose if you have certain plants, from that you take the embryo and you take the directly, you take that embryo and do tissue culturing. So, you will be getting the plants you will be getting. So, here, what you have to do is you have to learn certain things in this. So for certain type of plants, they do the rescuing of the embryo and how I have to explain you, you have polyploidy. In the polyploidy, you have two different types of ploidies or two different distant related plants ఉంటాయన్నమాట. So a distant related plants, you do the crossing అన్నమాట. After doing the crossing, you will be getting it. But it may not give the plant, it may not grow into the plant అన్నమాట. So అట్లాంటి వాటిది after the fertilization when the embryo is formed, you take that embryo, you rescue that embryo. ఆ embryoని నువ్వు కాపాడేసి you take it and you put it in the tissue culture condition. See, this fellow is telling you what they are telling. Embryo culture developed from the need to rescue embryos from the wide crosses where fertilization occurred, but embryo development did not occur. Wide crosses, for example, నేను మీకు మనకి మన ఆర్గానిజమ్స్ లోపలనే చెప్తాను, నేను హ్యూమన్ బీయింగ్స్ లోపల కూడా యు ఆర్ హ్యావింగ్ ఇన్ విట్రో ఫర్టిలైజేషన్స్ అనేసి ఐవీఎఫ్ బేబీస్ అనేసి అని అంటారు అనమాట. సో వాట్ దే డూ ఆఫ్టర్ ద ఎంబ్రియో ఫార్మేషన్, పిండము తయారైన తర్వాత యూ పుట్ ఇట్ ఇన్ టు ద హ్యూమన్ బీయింగ్ మదర్ వూమ్ లోపల పెడతారన్నమాట. సేమ్ లైక్ దిస్ వైడ్ క్రాసెస్ ఉంటుంది, అంటే అన్రిలేటెడ్ ప్లాంట్స్ విత్ క్రాసెస్ ఉంటాయి అనమాట. సో ఇన్ దోస్ ఆర్ related only, ఒకటి wild variety or one is domesticated variety. If the cross is done in between both of them, you will get the embryo formation. But that embryos will not grow into the plant anamata. So padipodamo, leaf develop gaakapodamo, something or the other it will get dried up. Atlanti what is after the embryo is formed, you rescue it and you take it to the tissue culture and you will get the new plant anamata. Next, this embryo culture can further be developed for production of plants from embryos developed by non-sexual methods. What is this? Non-sexual methods, I told you. Tissue culture itself is somatic cells. అంటే మనకి somatic cells లో మాత్రమే, vegetative cells లో మాత్రమే మనకి జరుగుతుంది అనమాట. మనకి యూ డోంట్ హ్యావ్ జర్మ్ లైన్ సిస్టమ్ ఇక్కడ మనకి ఇన్వాల్వే కాదు, మియాసిస్ ఇక్కడ మనకి ఇన్వాల్వే కాదు. హియర్ you must keep that in mind. Okay, so here what they are telling from non-sexual methods. ఇప్పుడు హ్యాప్లాయిడ్ ప్లాంట్స్ ఉంది, పోలన్ గ్రెయిన్ తీసుకోండి, ఓవ్యూల్ తీసుకోండి. యు ఆర్ హ్యావింగ్ ఓవ్యూల్ కల్చర్, యు హ్యావ్ యాంథర్ కల్చర్, యు హ్యావ్ పోలన్ కల్చర్. Anther is what? Anther or pollen grain is what? It's a male gamete. That what you are doing your doing the tissue culture. It is only single. It will have only, it is gametes cavity, it will have only one set of chromosomes only it will have. Ovule only. Ovule lopala already you have the meiosis అయిపోయి ఉంటది, శాకరణ విభజన అయిపోయి ఉంటుంది. సో దాంట్లో మనము ఇఫ్ యూ డెవలప్ ఫ్రమ్ దట్, అందులో కూడా యూ విల్ బి హ్యావింగ్ ఓన్లీ హ్యాప్లాయిడ్ సెట్ ఆఫ్ క్రోమోజోమ్స్ మాత్రమే మనకి ఉంటుందన్నమాట, ఏకస్థితికమే మనకి ఉంటుందన్నమాట. సో అట్లాంటి హ్యాప్లాయిడ్ ప్లాంట్స్ యూ డూ డబ్లింగ్, అండ్ ఆఫ్టర్ డబ్లింగ్ ఇట్ యూ టేక్ దట్ ఫ్రమ్ దట్, యూ టేక్ దట్ అండ్ యూ డూ ద టిష్యూ కల్చర్ అనమాట. సో యూ విల్ బి గెటింగ్ ద న్యూ ప్లాంట్స్ యూ విల్ బి గెటింగ్ అనమాట. ఓకే, సో సేమ్ అదే, you take the embryo, you put that in the medium, from that you will be getting the plants manaki vastundannamata. Okay, next haploid productions. I told you embryo rescue, you will be getting the what do you call haploid plants, or anther culture. Through the anther culture or ovule culture you will be getting haploid plants only, because anthers are the male gametes, they represent the male client, and the ovules they represent the female client. అందుకని దేర్ యూ విల్ బి గెటింగ్ హ్యాప్లాయిడ్ ప్లాంట్స్ ఓన్లీ, బికాస్ ఆఫ్టర్ మియాసిస్ శాకరణ విభజన తర్వాత మనకి అది ఏకస్థితికం అయిపోతుంది అనమాట. మళ్ళీ ద్వైస్థితికం కావాలి అంటే ఇట్ రిక్వైర్స్ ఫర్టిలైజేషన్ ఇస్ రిక్వైర్డ్. సో హియర్ నో ఫర్టిలైజేషన్ ఇస్ రిక్వైర్డ్, బట్ from anther culture also you will be getting plants. Or plants మనం you call that as haploid plants anamata. Ovule also, from the ovule culture also you will be getting the haploid plants you will be getting. Plants will be produced, but seeds will not be produced after doubling the chromosomes also. So here you will have the advantage of producing seedless variety grape, seedless variety of watermelons. Itlantivi all this can be exploited from this haploid productions. So we will see this bulbosum method of this haploid production. So how they have rescued the embryo. So they have taken two things. One is Hordeum vulgare, and this is barley anamata. So one is a wild variety. Ante manamu adividi wild variety ante adividi first variety edaithe undo adavi jatidi, it is that wild variety. So దాని తర్వాత now we are domesticating కదా మనము అంతా. సో we have domesticated అవి మనం we are eating that అనమాట. సో the wild variety is Hordeum bulbosum and this is Hordeum vulgare. So they have done crossing it between these two చేస్తారన్నమాట. So when they crossed in between these two, naturally fertilization అంటే pollen and ovule fertilization has taken place, and you had embryo formation అయింది. But the embryo was not growing anamata. The embryo was not giving the new plantlet anamata. So what they have done, they have done the embryo rescue chesaramata. So in this embryo rescue, what they have done is both are 2n, but here they have eliminated the chromosomes of this wild variety. They have eliminated this chromosomes of this wild variety. Hence 14 has become 7. And దీని what they have done is they have done the doubling, they have done chromosome doubling they have done, and they have grown this plant and they have created a haploid barley plant they have created anamata. So appadi nunchi from that time you call this method as bulbosum method. So eliminating one set of chromosomes and rescuing the embryo, and you get the plant. So that method they have called as bulbosum method. Okay, then next it can be anther or microspore. It is a male gamete. Okay, so same, you take the pollen grain. So this is the anther. So anthers is తీసుకొని you directly culturing the nutrient media. So it is two types. I told you always tissue culture two types. Direct organogenesis కావచ్చు, or through callus you can get anamata. So from this you can get callus. Through callus you can get the haploid plants, or directly from the anthers you can get the somatic embryogenesis. From that, you can get the haploid plants. But whatever it may be, from the embryo rescue and from this anther culture or the ovule culture, you will be getting only the haploid plants. Why only you will be getting haploid plants is these porins and ovules as such, they are haploid. అవి ఏకస్థితికం అనమాట. దే ఆర్ గ్యామెట్స్ అనమాట. దే ఆర్ నాట్ ఇన్ డిప్లాయిడ్ కండిషన్స్ కాబట్టి యూ విల్ బి గెటింగ్ ద haploid plants only you will be getting. Okay. So what are the anthers and anther or microspore culture factors? Then genotype plays an important role for all, of course, for all the tissue culture plants and matter. Next, selection of the mother plant also. Certain growth conditions for each mother plant. For example, potato has one type of culture. Coriander has one type. Carrots has one type. So each mother plant, it has its own growth conditions that we have to see. Then we have to see pollen also, at which stage that pollen is there, that also we have to see, but whether the pollen has just formed or whether it is turning into the pollen tube, it is forming, or which stage it is, that also correct stage of the pollen grain has to be taken. Then we have to see, we have to give certain pre-treatments for the anthers. It can be cold treatment or it can be the heat treatment. Then again, culture media also has to be very, very important. So selection of culture media, whether it's a liquid culture or broth or whether it is solid media, that also we have to take into consideration. Same goes with the ovule also. Ovule also same. You take the ovule and you put the thing, you put it in the medium. From that you will be getting the plants you will be getting. Usually in the ovule, it is very, very easy for ovule culture in onions and in melons and matter. So what do we do with the haploid plants? So first is the haploid plants, they are weak, but they are sterile. Sterile ante na microorganisms free కాదమ్మా. Sterile ante the sterile ఇక్కడ అంటే వంధ్యత్వము. That means it will not be able to produce the seed setting. అది produce చేయలేదు. ఎందుకు produce చేయలేదు all haploid plants? Let it be anther or let it be the ovule, it will be having only a single set of chromosomes ఉంటుంది అనమాట. When the seed setting will form, when you will be having two sets of chromosomes, that two heterozygous, two sets, one from the mother, one from the father ఉన్నప్పుడే you will be having the diploid condition lo you will be having the seed setting anamata. The first thing is, the drawback here is it is weak. The haploid plants are first of all weak and it is sterile in nature. Then usually what you have to do is we have to do the doubling of the chromosomes. So doubling of the chromosomes is called as colchicine treatment ఇవ్వాలా. Colchicine treatment is here what will happen is the doubling of the chromosomes will take place, but mind you, you can't call them as diploid. You can't call them as diploid. Diploid అంటే ఏంటి? Diploid is one is coming from the father, one is coming from the mother. ఆ రెండు heterozygous conditions ఎప్పుడైతే మనకు అక్కడ ఉంటుందో that is called as a diploid. కానీ this haploid same దాన్నే you're doubling it. ఆ haploid దాన్నే you're using colchicine and you're doubling it. That's why it is called as dihaploid. That's why you call that as dihaploid, but not diploid. Adi, you have to keep that in mind anamata. Haploid plants, if you give colchicine treatment when doubling take place, then ఏమంటారు అనేసానంటే diploid అంటారు, and if don't write it because it is not diploid. You don't have heterozygosity there. You're not getting that one from mother and one from father. You're getting this from the same haploid. దాన్నే డబుల్ చేస్తున్నారు కాబట్టి you call that as dihaploid plants with normal growth and fertility avanta everything will be normal, but it is dihaploid. So the chromosomes can be doubled by colchicine treatment, or sometimes spontaneously also you will be having the doubling manaki kanipistu untundi anamata. సో సార్ ఇది స్టాప్ చేయమంటారా లేదా ప్రోటోప్లాస్ట్ కూడా చెప్పేయమంటారా సార్? హలో
మేడం, ఈ రోజు ఆపేద్దాం.
ఓకే, ప్రోటోప్లాస్ట్ చెప్పేస్తే మళ్ళీ వీళ్ళకి ఎక్కువ ఇదే అయిపోతారేమో అనేసరని. వెన్ షల్ వి హ్యావ్ ద నెక్స్ట్ క్లాస్ సార్? We'll start with the protoplast, sir, next class. My dear students, if you all are not understanding, you can please ping me, or you can ask me again questions. I will reach out to you all. I can explain you in Telugu also. Please feel free. Okay,
Okay, ma'am.
sir. If this for today's class, if it is completed, we will stop here, sir.
Okay, madam. Thank you very much.
Sir,
You have wonderful presentation, madam. Excellent class, madam. Thank you.
thank you, sir. Thank you so much. Thank you, students, for joining me. And feel free, if you have any doubts, you can reach out to me. Thank you, sir. Thank you.
Thank you, madam. Thank you. Tomorrow
Okay.
we will